So you definitely have your packaging tomorrow. I'll give you a little bit of a jump on and kind of move forward. But we need to talk about a few things here. You're definitely welcome to have your packet open. You can kind of be taking some things down as we go if you haven't gotten it. Of course, you've got some of this stuff. Or you can just kind of look, whatever you want to do, kind of taking notes. But definitely kind of try to stay active. So we're looking at different types of cells. We talked a little bit about prokaryotes, eukaryotes. Here's a nice diagram of a prokaryotic cell. A couple key structures that we should be familiar with. You might want to kind of check these because this is a diagram you might have to label on a possible quiz or a test. Right here is the bacterial chromosome. Bacteria tend to have one kind of large circular chromosome, looks like that. And it's located in a region known as the nucleoid region. It's not a nucleus, right? There's no membrane around it, but it's kind of the nucleoid region. A couple of things we should be familiar with here. We definitely should be able to recognize the plasma membrane. We definitely should be able to recognize the cell wall, and we should be able to recognize the capsule on the outside. The capsule can be a protein coat sometimes. That's like an additional protective layer for bacteria. So bacteria not only have a plasma membrane, not only do they have a cell wall, they also have this outer coating known as a capsule. So those are kind of key structures that we should be able to identify. And it goes from plasma membrane out, plasma membrane, cell wall, capsule. Just think, the newest thing goes on the outside. We always know cell walls are outside the plasma membrane with plants and so forth. For the most part, that's kind of what we need to be able to identify. These longer tails are known as flagella. Just kind of a general thing to know. That's not necessarily specific to bacteria. There's other cells like that. And these fimbriae are kind of these small hair-like projections. We could also call them cilia, would work as well. Okay? So just looking at that diagram, I'm just going to highlight those are the key things that we should be able to identify. Again, prokaryotes are bigger or smaller? Smaller. And what are the three key features of prokaryotes? If I would have three identifying things of prokaryotes, what would I use to identify them? And we just said the first one. They're much smaller. Another one. They are lacking a nucleus. And the other key thing is they do not have, they have organelles, but they don't have membrane-bound organelles. They do not have membrane-bound organelles. Very good. So those are kind of the three key features of prokaryotes. Eukaryotes, we got a lot going on here. We're not necessarily going to label this one right now. But what are the three key features of eukaryotes compared to prokaryotes? They're much bigger, right? They're larger. They're typically about 10 times larger than prokaryotes, at least 10 times larger than prokaryotes. What else? They have a nucleus. They're more complex because they have a nucleus with their DNA package, and they also have these membrane-bound organelles, the membrane-bound organelles. So those are kind of key features that we should be familiar with. So they're much larger, they're more complex, they have a nucleus, they have membrane-bound organelles, even more than three, but they all kind of work together. Okay. One of the first structures we take a look at, we just mentioned it briefly because we have a whole chapter on it, you're doing a whole project on it, is the cell membrane. Notice the cell membrane, is it just made of those phospholipids? No, a common term used to kind of describe the cell membrane. First of all, it's a phospholipid bilayer. So there's two layers of phospholipids. Remember, lipids have the hydrophilic heads, the hydrophobic tails. By having two layers, the tails can face inward, protect themselves from the cytoplasmic fluid and the extracellular fluid, and the hydrophilic head can point outward. So it's a bilayer. But the other word we use sometimes, or the way we talk about our cell membrane is the fluid mosaic model. When I use the word mosaic, what does mosaic art look like? When somebody's doing mosaics, what does that type of artwork look like? Oh man, Mr. Snow pretty upset. Yes. Yeah, like little stones, little fragments. A lot of little smaller fragments that can be used to kind of create a picture. You ever seen the online, it's kind of a mosaic online as well. You ever seen the online ones where it's a bunch of smaller pictures, very tiny pictures that are used to kind of make a bigger picture? Sometimes if someone's face and a bunch of smaller pictures of the person, and they use it for the big one. Mosaics with stones or glass are usually small little fragments. They kind of piece them together to make an artwork, an image. So we use the word mosaic here because it's a mosaic of not just phospholipids, but we also have these proteins, these carbohydrate chains, which are oligosaccharide chains, and so forth. So there's a lot of other components here. We're not going to spend too much time on the membrane right now because we'll spend more time on it later. This is kind of a cool little picture. Here's a TEM of a plasma membrane. What am I saying when I say TEM? Transmission electron microscope, right? We can get a nice clear picture, and it's pretty amazing that, look at, not only are we seeing the cell membrane, we actually have enough resolution, right, with the TEM to actually differentiate the two layers of our membrane, the upper layer and the lower layer of the lipid bilayer. Amazing. Mind blown. You've never seen a picture of the lipid bilayer before. Amazing. It's just a great diagram. I will go back to this later when you're not going to have a quiz this week. Well, I might have something next week or something. But yeah, a quiz will be a nice little labeling quiz. Here's a good one. We'll go back to practice with this later. And here's kind of this one would be what type of cell? Eukaryotic, but also animal cell. This one's eukaryotic, but a plant cell. Another thing we might have to label, right? So these are kind of good practice ones. Again, I will go back to this later once we move through some of the organelles. And this is a good one to label. And this would be a prokaryotic cell, bacteria, and so forth. So we will go back to these and practice a little bit as we move forward. Okay, what I want to do is I kind of want to take you on this tour of the cell. Just a little video. It's on YouTube, so you can see it online. You'll also be able to see it here. Escape. There we go.
Even if you're just studying, a lot of work is going on in your cells. Let's zoom down and check it out. We've arrived in the space between two cells.
Okay, there's a lot going on in this video. So you're going to kind of get this whole big picture of a cell, all these different components going together. So I'm going to stop here and there to make sure to ground you, to make sure you understand what you're looking at. So what we're looking at here, we are not inside a cell yet. We are outside the cells. We're in between cells. So here is the upper layer of the bilayer of one cell, phospholipid bilayer. Here is another cell. What we're looking at, again, I don't know if we've quite gotten to the path yet, but we're looking at the matrix that's outside of the cell. Cells are connected to one another, and they're connected to what's known as the extracellular matrix, the ECM. So what we're looking at here is the ECM, the extracellular matrix, the carbohydrate, the protein fibers that help to kind of hold these cells together, to allow these cells to communicate with one another, to identify one another, and everything. The purple things, if the yellow or the brownish are the phospholipid bilayers, what are all these purple things? Not ribosomes. Ribosomes are going to be inside. Carbohydrates could be these little chains coming off. The purple is given away with the P. Not plasma. Proteins, right? The purples are proteins. They could be just proteins that are just embedded there. They can be protein channels to help move things through. Right? Proteins can serve different functions on the membrane. One of them is going to be a protein channel, which you'll see in a second because we're going to go through it.
A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Wow, look at this place.
Those are just molecules that either possibly for communication. So they could be hormones or just chemical messengers where one cell is communicating with another cell. They could be possible food sources, maybe nutrients that they're going to be using, maybe a glucose molecule. There's a number of different possibilities of what they could be. But cells communicate with one another, and cells also are constantly taking in material, so it could be a food source. Okay, so now we just went inside the cell. It's a mess. Look at this, right? There's all this cytoskeleton inside, right? All our cytoskeletons, the microfilaments, the microtubules, the intermediate filaments, right? Notice we got a little mitochondria down here, right? The little yellow things coming off the mitochondria is going to be energy. What was our energy molecule? ATP. Those little ATPs coming off. Little vesicles. You're going to notice this cytoskeleton. It's not just this static structure. It's not just the structure that holds the cell and gives it rigidity and shape. That's one of its functions, to kind of hold the shape of the cell. But the other function is it's really a transport system. It's like the railway system of the cell. So it allows it to move things around. So you kind of see how it operates here.
These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made.
Yeah, kind of powerhouse. A couple of key things there. One is there's two membranes. It's a double membrane structure. It's the inner membrane and the outer membrane. We'll talk a little bit more about the importance of that when we deal with cellular respiration in the mitochondria. One thing is that inner membrane has all the folds. And in biology and science, if I want to increase the function of something, oftentimes I can increase the surface area. So that's like a big concept to understand. We'll talk more about that, but it's definitely key to know that the mitochondria and the chloroplasts are double membrane structures. Okay? And we'll talk about why later on.
Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus.
Did that just blow your mind? Do you know what I'm talking about? What am I talking about?
2 meters.
2 meters. How much is 2 meters? 6 feet. There's a length of 6 feet of DNA in every single one of your cells. That's a lot of DNA. Now, how do we get in? It's very fine, so it coils and it wraps and it coils and wraps, kind of keeps it all together. But think about how much DNA is in each one of your cells. That's a tremendous amount of DNA. You're seeing the wrapping here. Do you see the double helix? Does anybody see the double helix up here? No, we're not down to that structural level. We're at a lesser magnification, we'll say. So what you're seeing is these strands right here, that's the DNA molecule, and you could get down to the double helix there, but you're not really going to see it. The purple things are proteins that the DNA wraps around, and those proteins are known as histones, which we'll talk about later as well, but histones. Does this DNA look readily accessible to get information out of it? No. Does this DNA look more accessible? Yeah. DNA, go ahead. Not small, it's unwound. They're the same thing, but in the bottom it's been wound up, packed tight together, and in that section there it's unwound a little bit. So there's kind of this large control of our DNA of winding and unwinding it, which plays a major role with our with whether traits are expressed or not. Does anybody know the molecule? Again, we're not talking about DNA genetic. Does anybody know the name of the molecule that copies DNA? Let's do this. It's a good time to kind of introduce a big concept for biology again, to kind of see how all these cells function. I think I mentioned it before, but what is the central dogma of molecular biology? Don't Google it. It sounds like a really confusing thing, but you already know it, right? It deals with DNA, RNA, and proteins, and it's simply a progression. Does DNA ever leave your nucleus? No, we don't want DNA to leave nucleus. DNA is very important, correct? So we don't want it to leave our nucleus. We keep it in the nucleus. But ribosomes, what's the job of a ribosome? To make proteins. Do ribosomes go in the nucleus? No, so how do ribosomes get the instructions to make proteins? The DNA holds the instructions, and they're in the nucleus. And ribosomes are in the cytoplasm. How do they communicate? Messenger RNA, so RNA. We'll talk more about this, but this is a big concept that kind of goes through the operation of a cell. So the central dogma is simply DNA to RNA to proteins. Okay? All right, so that's what you're going to see here just a little bit. Just a little bit. What I'm trying to get you to is oftentimes the cell chapter is simply just a memorization of mitochondria to the powerhouse. This is the headquarters. The Golgi apparatus is the packaging center. What I'm trying to have you see is this is really an entire network. They all work together in conjunction with one another. This is a machinery. There's a mechanism here that allows cells to operate. All right, so we're looking at DNA right now.
How does it all fit? The DNA is wrapped around proteins, like thread wrapped around spools. Look, this section of the DNA has unwound, and a different protein has attached to the DNA. DNA is being used as a template.
The red stuff is
mRNA.
RNA.
Molecules travel from the nucleus to the cytoplasm.
How does it get out of the nucleus? What does it pass through? The pores, right? The nuclear pores. Very good. And now you're going to see it's leaving the nucleus, going through a nuclear pore.
Carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new.
All right, now it's making what? That little purple thing coming off is a? No, the brown thing's a ribosome. It's reading the RNA. The purple thing coming off is protein, right? That's the job of ribosomes, to make proteins. Okay? All right. We don't just have one type of ribosome. We have two types of ribosomes in our cell. What are the two types of ribosomes? Really dealing with their locations. Free and bound, or attached. Obviously, what are we looking at here? This one is free. It's a free ribosome, which means it's just floating around in the cytoplasm. That tells us something very important. I would just jot kind of these things down as a thing. That's very important because when the ribosome is free, the protein it's going to make is most likely going to stay intracellular. It's going to stay inside the cell. So free ribosomes make proteins that, for the most part, stay inside the cell. Make proteins that stay inside the cell. Everybody okay?
Protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are two types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can...
Okay. So, I'm going to talk a little bit about what the endomembrane system is. Right? That's a term you might have heard, you might not have heard before. It's just a big word for saying, hey, guess what? All these membrane-bound organelles, they all kind of work together. They're their system. And the key ones are the smooth ER, the rough ER, the Golgi apparatus, the nucleus, the cell membrane, and all the little vesicles in between. They're all membranes, which means they're all made of the same stuff, which means they're all made of what macromolecule? Lipids. That's why we call it the endomembrane. They're all the same thing. They carry out different functions, but it's all made of the same stuff. Okay? There's a progression. Which component of the endomembrane system's job is to basically make more lipids, make more membrane? So we have a few different ones here. We have the Golgi apparatus, we have the rough ER, the smooth ER, the nucleus, we have vesicles, the cell membrane. Which one's job? It has a number of different jobs, but one of the key jobs is it synthesizes lipids, it makes lipids. Yeah. The smooth ER, very good. Now the smooth ER is located pretty close to the nucleus and in close conjunction to the rough ER. So the smooth ER is constantly making more lipids, making more membrane. That's good. That membrane can shift right to the rough ER. Okay. So what we see here, just keep that in the back of your mind. What we see here is we're looking at, we're inside the rough ER. It's kind of hard to kind of visualize, but we're actually kind of like if you were laying at the bottom of the pool and you're looking up at the surface of the pool, that's what we're doing now. We're inside the rough ER looking at the membrane. The RNA is outside, so if you're looking through the surface, the ribosome's on the outside, but the protein that's being made is being made by the ribosome and going directly into the rough ER. Okay? What's the main function of the rough ER? It modifies what? Rough ER, main job, main function. Rough ER. R-O-U-G-H. R-O-U-G-H. And then endoplasmic. ER. Just go ER for that. Endoplasmic. What's the function of the rough ER? Come on, you should know this. You've been doing this for homework. You should have gotten at least this far. Jerry. That's the smooth ER. The smooth ER makes membranes. Mila. Okay, all those are functions, but there's really one thing I really want to boil it down to simplify it a little bit. Yes? It makes transport vessels, it definitely does, but what does it do to proteins on the inside? It modifies proteins, right? It modifies proteins. So what happens, right? This protein was just made by the ribosome. It goes into the rough ER. The rough ER inside can help modify these proteins, help to fold them properly. Maybe if you go from tertiary to quaternary, who knows? Okay, we're following proteins. Where do proteins go next? If they're modified in the rough ER, where could a protein go next? What organelle could it go to next? Typically, not all the time, but typically progression. If that protein was modified in the rough ER, it's going to go where? To the Golgi apparatus. Okay, right? Your cells are making millions and millions of proteins. Proteins are highly reactive. They're unstable because they can move around. They're very delicate. I don't want to send proteins by themselves. I need to protect them. So how do I ship the proteins from the rough ER to the smooth ER? From the rough ER to the Golgi apparatus? Don't answer that yet.
Still see the ribosome on the outside surface.
So there's our proteins getting
The
formed.
ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from.
There you go. It's an endomembrane system, so it's all just membranes. So the lipid membrane that was made in the smooth ER can help increase the size of the rough ER. The rough ER, as it's trying to work with the Golgi apparatus, has to keep sending these vesicles. What are the vesicles made of? Lipids, right? They're lipid membranes, right? That's why we keep needing more membrane, because we're constantly sending vesicles to the Golgi apparatus. Now, inside the vesicle are proteins, right, that are going to be used to carry out some function. That's why it's considered an endomembrane system. They all work together with one another. So how do they transport? That's where the cytoskeleton comes in. The vesicle will bubble off, and it will join onto like the railroad tracks of the cytoskeleton, and other proteins will help move them along. The transport. So they're going to be moved like a train track. Yeah. So those vesicles are going to go
The ER membrane.
to the
Some vesicles
Golgi apparatus.
The Golgi.
Notice, the vesicles can just be absorbed into the Golgi apparatus, because the Golgi apparatus is simply made of the exact same stuff. It's just membrane, so it's absorbed in. Those proteins that were initially modified in the rough ER now get absorbed into the Golgi apparatus, where maybe further modifications can happen. So I modified the proteins initially, but maybe I need to add some extra carbohydrate groups or do some extra additional things to make them completely 100% functional. So they're kind of now just further modified in the Golgi apparatus. What's kind of the basic term we use for the Golgi apparatus? The function. It's the packaging center, right? The Golgi apparatus is going to get things ready to ship it out of the cell. So once these proteins are further modified in the Golgi apparatus, they're then going to be packaged into another vesicle and sent to the membrane.
The apparatus. Another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
So just to kind of pull it all together, right? Your whole cell's real job is to make proteins, right? That's what we are. We're walking chemical reactions. We need the enzymes to function. If the ribosome is free, it's going to make proteins that are going to stay basically inside the cell for the most part. If the ribosome was attached to the rough ER and it's part of the endomembrane system, the proteins that are made are going to be modified in the rough ER. They're going to be shipped to the Golgi apparatus to be further modified, and most likely they're going to be shipped out of the cell. An example of this would be something like insulin. Insulin helps. It's a hormone that helps to control your blood sugar levels. Right? When you have a lot of sugar, your body secretes insulin to help take sugar out of your blood. Well, the cells secrete the insulin. So you can picture the cell making more insulin and then shipping it through from the ER to the Golgi apparatus to the cell membrane and releasing it into your bloodstream. Okay? So hopefully I give you a little bit more of a picture of kind of how this system works. We're trying to get away from just memorizing individual components of the cell and seeing how they actually function together as like machinery. Any questions? Can I ask one question? Did this help you get a better understanding of the cell? Or are you like, what the heck are you talking about? Because I had this nice little understanding and now I don't understand what's going on. You go either way. How many people feel like they have a better understanding of the cell? How many feel like this confused you more? You can be honest, it's okay. All right.